Hello, my name is José Leme, and first I would like to thank you for participating in this conference, even with all the changes and these new normal settings, I guess. And obviously, I would prefer to be traveling to Arizona, but you know, we have to play by the rules. It is what it is. So I'll be talking about our study addressing the use of biostimulants on cannabis under outdoor conditions. This study is uh, an extension of a greenhouse study that we have conducted previously. And based on the promising results, we decided to extrapolate it to outdoor conditions. Also, it's an ongoing project, so I will be presenting only the data, the preliminary data from 2019. The main approach of this study is the integrated plant nutrition system. And the fertilizers derived from inorganic, organic, and biological resources are different in terms of ability to supply nutrients, rates, soil fertility, and plant growth. And the integrated plant nutrition system definition is basically the combination of inorganic, organic, and biological nutrient resources in the most efficient way, taking into consideration the environmental, social, and economic reality of the location or of the area. So for this current project, we use NPK as an inorganic resource, so it's a no surprise. As an organic resource, we use humic acid and manure tea, and as a biological resource, we use manure tea and bioinoculant. Um, so manure tea, it's considered organic and biological resource. So something that is very important to clarify right here is that manure tea and bioinoculant are under the category of biofertilizers. So every time that I'm talking about biofertilizers, I'm referring to manure tea and bioinoculant. Something that is also very important to mention is that humic acid, manure tea, and bioinoculants are under a general category of biostimulants. So again, every time I'm talking about biostimulants, I'm referring to all the products, humic acid, manure tea, and bioinoculant. So the hypothesis of this study is that synergetic effects from the combination of biological organic resources and inorganic fertilizer could result in improved agronomic outcomes. So now we might be asking, under the category of organic and under the category of biological, there are several options. There are so many different products that we can use. So why you decide to specifically use humic acid and biofertilizers? Here's why. Studies have shown that the humic acid stimulates microbial activity as an important component in exchange and metal complexing chelating systems. Also, it increases the production of mycelium in mycorrhizal fungus. So basically, the rationale behind that is that humic acid will increase microbial activity in the biofertilizers, and the overall performance will be maximized. So our objectives are evaluate the effects of NPK, humic acid, manure tea, and bioinoculant as inorganic, organic, and biological resources and their synergy on cannabis growth and soil indicators. Also define if the combination of humic acid plus biofertilizers is beneficial based on the plant and soil parameters. So with this slide, I just would like to clarify that among all the treatments, 2 treatments received only biofertilizers, 1 treatment received only humic acid, and 2 treatments received humic acid plus biofertilizers. So, and obviously the control. And The inorganic fertilization followed the tomato protocol for nutrients requirements. So I just a very quickly background of the products that we use in this study. So humic acid can be extracted from any material containing well decomposed organic matter as soil, coal, peat, compost, and leonardite. The microgel, which is the minority, is a patent product from a Brazilian company. And in order to make the final product to be applied on crop, we need a tank. Then we add the manure inside the tank and the microbial food and water. So, and then after 15 days of an aerobic fermentation process, the final product is ready to be applied on crops. The MicroGrow Supreme Bioinoculant, it's basically a water-soluble powder containing several beneficial microbes and microbial food. So, and in order to make the final product, we have to dilute this powder in water, and then the final product is ready to be applied. So, some technical information about this study. We use 6 feet row spacing and 5 feet plant spacing and 6 plants per plot. So this spacing is very common for CBD production. Total of 4 treatments application, 2 drench application, and 2 foliar application. We also collected plant high, at-leaf chlorophyll meter, fluorimeter, which is the photosynthetic efficiency, vigor and greenness at 13 and 17 true leaf pair, and 2 weeks after flowering. The above ground plant materials were harvested for biomass yield. Soil samples were collected after harvest. And we used randomized complete block design with 4 reps. Okay, finally, the results. So we have 3 graphs here: cannabis high at 13 leaf pair, 17 leaf pair, and 2 weeks flowering. Y-axis, high in centimeters, x-axis, the treatments. I also would like to use these graphs to provide you some information that I will be using for the coming bar graphs. So as you can see, the white is referred to control. The dark blue bar is the humic acid treatment. The medium blue bar is the biofertilizers or the biological resources. And the light blue are the combination of humic acid plus biofertilizer. So I use the same approach for the coming bar graphs. So when you see, but I use different colors. So if you see dark purple, medium purple, and light purple, so it's so on. So it's the same approach. So specifically talking about cannabis high. So high presented statistical difference in only one data period, as you can see the second graph, 17 leaf pair. And humic acid treatment showed the highest value when compared to the control and the other biostimulant treatments. So at 13 leaf pair and 2 weeks after flowering, no statistical differences were found. So chlorophyll content. We use the at-leaf divide to collect the chlorophyll content and to measure the chlorophyll content. And so in this graph, y-axis, chlorophyll content in milligrams per centimeter square, and x-axis, the treatments. Chlorophyll content present a statistical difference at 17 and 2 weeks flowering. As you can see, the first data period, 13 leaf pair, did not show any statistical differences. However, the control present the lowest value at 17 leaf pair and 2 weeks after flowering. And the no treatment stand out among the biostimulants treatments. So In other words, no treatments showed higher values when comparing to the treatments receiving humic acid and or biofertilizers. So photosynthetic efficiency, we use the fluorometer to measure the photosynthetic efficiency. And again, in this graph, photosynthetic efficiency, x-axis, the treatments. And similarly to the chlorophyll content, the photosynthetic efficiency presented statistical difference at 17 leaf pair and 2 weeks leaf pair in 2 weeks flowering. Again, the first data period at 13 leaf pair did not show any statistical differences. And again, biostimulant treatment stand out when comparing to the other biostimulant treatments. And these two plant parameters might be correlated because one parameter is measuring chlorophyll amount in the leaves, and chlorophyll is correlated to photosynthesis. And some studies are showing this correlation, but unfortunately I don't have time to go deeper on it. But it's that might be one of the explanations why these two parameters are somehow similar. So finally, the biomass yield. So two graphs right here, cannabis total biomass and cannabis bucket biomass. If you don't know what is bucket biomass, it's basically the most common way that cannabis farmers commercialize their CBD production. And bucket biomass is the total biomass minus the stem. So it's just no stem. The stem is out of the picture. So it's somehow two different ways to measure yield. So let's go deeper to the graphs. First graph, cannabis total biomass, y-axis, total biomass in kilograms per hectare, x-axis, the treatments. Second graph, y-axis, bucket biomass in kilograms per hectare, x-axis, treatments. Only one treatment receiving the combination of humic acid plus biofertilizer presented statistically higher values than the control and the most of biostimulant treatments. So again, these two ways to measure biomass, measure yield, are not exactly the same, but they behave similarly. And with that said, going back to our hypothesis, it closely approximated to our initial hypothesis, which is that the synergy of humic acid plus biofertilizer applied together would increase cannabis yield comparing to each of the products applied individually and the control. So again, in fact, the combination of humic acid plus biofertilizer presented the highest value. However, we test two treatments receiving humic plus biofertilizer, but only one present the highest value. Jumping to the only soil data of this study. So soil respiration, y-axis, CO2 production per grams of soil, x-axis, the treatments. Again, as occurred to the biomass, it approximated to our initial hypothesis where the humic acid plus biofertilizer would present better performance than control and the independent application of each biostimulant, as you can see here, right here. But again, among two treatments addressing the combination of humic acid and biofertilizer, only one presented significant difference. So these leave us with many opportunities and ideas for next studies to kind of clarify if this combination of humic acid plus biofertilizer is in fact better than the application of each one alone, independently. Some preliminary results, just to try to to explain